Hi guys, welcome to BAL 2165 Practical Tree. So this is the video presentation for your Practical Tree lab session. Important things to note, there will be 2 query sessions for this practical. One on Tuesday between 1 and 3 p.m. and the other on Saturday from 9 to 11 a.m. So pay attention to the video, and if you have any questions, attend the query session and your demonstrators will be there to assist you. Please note that all lab scripts, regardless of which query session you attend and which lab stream you belong to, will be due on Tuesday, 3rd of November by 4 p.m., since all necessary information will be presented to you in this video. What is bacterial conjugation? So your practical session today is based on bacterial conjugation. Conjugation is a mechanism by which DNA from one cell is transferred to another cell to produce a new recombinant cell. Sometimes the DNA that is being transferred codes for antibiotic resistance. This intercellular transfer of this bacterial DNA coding for resistance to antibiotics enables the new recombinant bacterial cell to express resistance to an antibiotic to which it was formerly sensitive. This transfer is also considered as a type of genetic recombination. While bacterial chromosomes normally carry all the genes necessary for growth and reproduction, bacteria also contain genes carried on extrachromosomal DNA called plasmids. Plasmids are double-stranded circular pieces of DNA that carries genes. Some of these genes regulate the formation of villi, which are elongated appendages that extend from the surface of the cell. These villi can function as a bridge between two bacterial cells, thus allowing the transfer of DNA from the donor to the recipient. Your lab procedure is divided into 3 lab days. Lab Day 1 involves the confirmation of antibiotic resistance in each bacterial strain, while Lab Day 2 involves observation of the confirmation plates and preparation of the mating plates. And finally, on Lab Day 3, there is observation of recombinant growth and donor determination. In preparation for Lab Day 1, pure cultures of E. coli strains 1 and 2 were grown in LB broth and enriched medium. As seen here, strain 1 E. coli cells contain the gene for streptomycin antibiotic resistance on the chromosome, while strain 2 has a gene for nalidixic acid resistance on its chromosome and a gene for ampicillin resistance on a plasmid. And what this means is that since strain 1 contains a gene for strep antibiotic resistance, it will therefore be resistant to strep antibiotic and will be able to grow in the presence of this antibiotic, while strain 2 contains resistance for 2 antibiotics, which are AMP and NAL, and therefore since it contains resistant genes for these both antibiotics, strain 2 will be able to grow in the presence of these antibiotics. On Lab Day 1, samples of each strain are transferred to LB agar plates containing the enriched growth medium and the antibiotics to which each strain is known to be resistant to. You will be provided with 5 LB agar plates as illustrated in this diagram. Plate 1 is plain LB agar. Plate 2 has the antibiotic streptomycin infused in its agar. Plate 3 has the antibiotic ampicillin, while plate 4 has a mixture of both antibiotics strep and amp, and plate 5 has nalidixic acid. Each plate is then divided into 2 regions, 1 and 2. Region 1 is inoculated with bacterial strain 1, while region 2 is inoculated with bacterial strain 2. This exercise confirms resistance or sensitivity of these strains to the antibiotics. Here, the video for Lab Day 1 is demonstrated. The 5 agar plates are laid out in the same order as seen in Figure 2 in your lab manual. The technician then proceeds to divide each plate into their respective regions. Region 1 is labeled with Roman figure 1 and region 2 is labeled with Roman figure 2. A line is therefore drawn on the bottom surface down the middle of each plate as seen here to divide the plates into the two regions. Before beginning the procedure, the bench tops should be sterilized using 70% ethanol. Please observe each plate. Bacterial strain 1 is contained within the centrifuge tube. The handle end of the loop's wrapper is opened and the sterile loop is then removed. The cap is removed from the strain 1 broth culture bottle. She then proceeds to dip the sterile loop into the strain 1 culture. A loop full of strain 1 bacterial cells have been collected, and she then begins the inoculation process. The loop is touched lightly to the surface of the agar in the middle of each section marked Region 1 on each of the 5 plates. At the end of the process, each plate is inoculated with bacterial strain 1 in Region 1 of the plates. The procedure is then repeated using bacterial strain 2. A second tube contains bacterial strain 2. Similarly, a sterile loop is used to collect a loopful of the strain 2 bacterial cells. Region 2 on all 5 plates is then inoculated with strain 2 bacterial cells. At the end of the process, Region 1 and Region 2 would have been inoculated with strain 1 and strain 2 bacterial cells respectively. Now that the process of inoculation has finished, it's time to seal the plates. Parafilm is used to seal the edges of the plates. The plates are then stacked, inverted, taped together, and labeled appropriately. The confirmation plates are placed in an incubator and left overnight to incubate at 37 degrees Celsius. After the incubation period, each plate is carefully observed to detect whether there was growth of bacterial cells, and these results are to be recorded in Table 1. So please take some time and pause the video accordingly at this point to observe the bacterial growth response. This is your results table for Lab Day 1. In Table 1, input whether there was expected growth or no growth of strains 1 and 2 on the plates based on what you know from theory. Therefore, consider the antibiotic genes of each strain and say what you would expect. For example, if you recall, strain 1 has the antibiotic resistance gene for strep on its chromosome. Therefore, you would expect to see growth on the LB agar plus strep plate. For the observe category, you will need to pause the video and observe each plate to determine where there was growth. The mating plate is prepared on Lab Day 2. The mating plate contains a mixture of LB agar, AMP, and strep antibiotics. The plate is divided into 3 regions as seen in Figure 3. A sterile loop is used to place a drop of strain 1 onto region 1 and onto the mating region of region 3. A separate loop is then used to place a drop of strain 2 onto region 2 and then onto region 3 about 1 centimeter away from the drop of strain 1. The loop is then used to mix strains 1 and 2 in region 3 and spread the mixed cells over the surface of the plate. The procedure for Lab Day 2 is now demonstrated. The workspace is first sterilized using 70% ethanol. Please observe the mating plate. The mating plate is a mixture of LB agar, strep, and AMP antibiotics. She then proceeds to divide the plate into 3 regions as illustrated in Figure 3. The areas on region 3 where she intends to inoculate strains 1 and 2 are also identified. The purpose of this plate is to bring the bacterial cells into close proximity with each other so that conjugation can occur. This tube contains strain 1 bacterial cells. A sterilized loop is used to collect a loopful of strain 1 bacteria. Region 1 is now inoculated with strain 1 bacterial cells, and some of these cells are also placed onto the area marked 1 in region 3. A clean loop is then used to collect a loopful of strain 2 bacterial cells. Here she inoculates region 2 and area 2 on region 3. Next, she mixes the strains together and spreads the mixture over the surface of the plate. The plate is then sealed with parafilm, inverted, and placed in an incubator at 37 degrees overnight. At this point, you should pause the video to observe the growth pattern on the plate. This is the results table for Lab Day 2. Indicate in Table 2 whether you would theoretically expect to see growth or no growth in regions 1, 2, and 3. Carefully observe the results for the mating plate in the video and indicate if you observed growth or no growth in regions 1, 2, and 3. In the box below, You should explain your reasoning for the expected results indicated in Table 2 with respect to regions 1, 2, and 3. For example, if you state that no growth is expected in region 1, in this box you should explain why. Remember that the mating plate contains a mixture of both strep and AMP antibiotics. Something to consider here is that even though strain 1 may be resistant to strep, since it has the gene for strep resistance on its chromosome, it will be sensitive to the other antibiotic, which is AMP. Please note that any growth observed on the mating plate will be growth of recombinant cells, therefore those in which conjugation has occurred. No growth should occur in regions 1 and 2 since each strain is sensitive to one of the antibiotics. The response of the recombinant cells from the mating plate to the different antibiotics is investigated on lab day 3. 4 different antibiotic plates are used as seen here. Plate 1 has a strep antibiotic. Plate 2 has a mixture of strep and AMP. Plate 3 has AMP antibiotic, and plate 4 has NAL antibiotic infused in its agar. Each plate is then divided into 5 sections using a marker as illustrated in the diagram. A single colony from the mating plate is collected and section 1 on each of the 4 plates is inoculated. The loop is sterilized and again a single colony from the mating plate is collected, and this time, the second time around, section 2 on each of the 4 plates is inoculated. This process is repeated until all 5 sections on all of the plates are inoculated with the recombinant bacterial cells. The procedure for lab day 3 is demonstrated here. Please observe the antibiotic plates. Here, a 5th plate is included, which contains no antibiotic, its plain nutrient agar. She then proceeds to divide each plate into 5 sections, as illustrated in Figure 3. Please observe how each of the plates have been divided into the 5 sections. 70% ethanol is used to sanitize the workspace. She then displays the mating plate from day 2. Note the growth of recombinant cells in region 3 of the plate. A sterile loop is used to collect a single colony of bacteria from the mating plate. The isolated colony is then transferred to section 1 of each plate. Notice how she heats and then cools her loop, therefore sterilizing it before she moves on. The process is then repeated. Another colony of bacteria is collected using the sterile loop from the mating plate and transferred to section 2 on each of the antibiotic plates. The process is repeated each time with a new colony until all 5 sections on each antibiotic plate has been inoculated with recombinant bacterial cells. Once completed, she then seals the plates with parafilm, after which they are stacked, inverted, and labeled appropriately. The plates are then placed in an incubator overnight at 37 degrees Celsius. Please attentively observe the growth results from each plate and record your observed results in Table 3 of your lab handout. This is your results table for Lab Day 3. Record your theoretically expected results in the form of growth or no growth for the growth response of the recombinant cells on the antibiotic plates. The expected results will vary depending on whether strain 1 is the donor or whether strain 2 is the donor and transfers plasmid genes only or chromosomal genes only. For example, using strain 1 chromosome as the donor, if strain 1 transferred the genes on its chromosome to strain 2 during conjugation, and as you'll recall, strain 1 has the resistant gene to strep, then this would mean that the new recombinant bacteria will therefore be resistant to strep, amp, and null. Therefore, if this is the case, we'll expect to see growth on all 4 plates. In the last column, your observed results are indicated. Pause the video and look at the results from day 3. Once there are bacterial cells or colonies seen growing in any one of the 5 sections or on all 5 sections of the plate, then this would be regarded as there being growth. Observe and record for all 4 plates. Once you've filled out all the expected results and the observed results, compare the expected results to the observed results, and whichever of the expected results the observed results matches, then that particular strain is the donor. To explain further, if these are your expected results in red and you have your observed results here in blue, upon evaluation, you would notice that the observed results matches the expected results in the first column. Therefore, we can conclude here that based on these results, strain 1 chromosome was the donor. Please note that these are arbitrary results used just for explanation, and it should by no means be interpreted as actual or real results. At this point, all results would have been collected, and you will therefore be able to answer 2 questions. By now, you should be able to confirm if conjugation occurred, and you should also be able to say which was the donor strain. And finally, there are 7 questions which you need to answer and discuss based on your results. Here are 2 of the questions which you need to answer. For example, question 1 reads, in the confirmation test, why did strain 1 not grow on the LB plus AMP plate? You should read the question carefully and make sure you understand what you're being asked. When answering these questions, you need to be very explanatory and give the theoretical reasonings for your answers. Recall that strain 1 has the resistance gene for strep. If a bacteria is resistant to a particular antibiotic, it means that it can grow even in the presence of this antibiotic. However, if a bacteria do not contain the resistance gene for a particular antibiotic, it will be sensitive to that antibiotic and will not grow, as the antibiotic will kill the bacterial cells. Recall that strain 1 has the resistant gene for strep. Therefore, it is resistant to strep. but here it is plated on media containing AMP, an antibiotic for which it does not have a resistant gene. Will it grow? And if not, then explain why. With respect to question 4, recall what antibiotics were present in the media on the mating plate. Did strain 1 and strain 2 have the antibiotic resistant gene for both these antibiotics? If not, it means that strain 1 and strain 2 would have been sensitive to at least one of these antibiotics. Therefore, with this in mind, you should be able to tell whether the growth on the mating plate consists of recombinant cells only. And this is just an idea on how you should approach these questions. Okay guys, I hope that you were able to understand everything. If not, please remember to attend any one of the query sessions and we'll be there to answer your questions. Also, you can feel free to email any of your demonstrators or your TA outside of these sessions if you need any assistance.